Good afternoon, everyone. My name is Alex Koon. I'm a senior lecturer of the School of Life Sciences at Chinese University of Hong Kong. 大家好，我雖然今日嘅 lecture 係用英文講啦 ，English 啦，但係其實大家如果係一陣問問題嘅話，隨時都可以變翻做廣東話，我係冇問題嘅，廣東話係 OK 嘅。只不過因為我平日教學都係用英文教嘅，所以一時間根本係冇可能將好多嘅 technical term 全部轉做中文嘅啫，係咁嘅意思，係啦。咁所以就不過應該都 OK 嘅。今日我嘅 lecture 都會盡量帶出嗰個深入淺出嘅精髓啦，希望就係一啲變咗，就算係冇讀過免疫學系嘅，都希望可以喺短時間之內 pick up 到呢個免疫學嘅一個 background 啦，然之後去理解究竟呢個 mRNA vaccine 同埋呢個 pseudouridine 有乜咁特別咧？咁樣樣嚇。好，我哋就事不宜遲啦。So let's get started. So today the talk of my title will be Psi, right? This is Psi, right? Pseudouridine, the U with an I that changed you and I. So we'll start off with a little bit of data about COVID-19. As we all know, this terrible disease has been bugging us for a couple of years at this point, right? Almost like 4 years already, nearly. And as we all know, the number of deaths has been rising at the beginning, right, until like 5 million or 6 million very quickly. Right now, the number is kind of dying down, hopefully, right, continue to go lower. But each year, there are still a significant number of deaths. And at this point in 2023, there is a cumulative of over 6.9 million deaths, right? And as we all know, COVID-19, right, what doesn't kill us will continue to mutate and tries again, okay, right? So as we can expect, COVID-19 is very resilient, right? It's going to mutate and it's going to come back again very soon. It's just like in this comic right here, right? This is the COVID-19 virus right here, represented by this Hydra. Like if you have seen the Hercules mythology, right? Greek mythology, you know, when you cut off a head of a Hydra, it comes back with two more heads, right? You can never kill it truly, but just by cutting off the head. And COVID-19 is just like this Hydra right here, okay? So at the same time, this comic also illustrates one thing, is the structure of the virus, right? If you know a little bit about the biology, you know that this is actually resembles the structure of a virus. Let's take a look. This is the COVID-19 SARS-CoV-2 virus, okay? It's very similar to that comic illustrated, right? So it has spike proteins and a number of other proteins, right? And doesn't matter because we are focusing mostly on the spike protein today, right? During infection, this spike protein, which is a surface protein on the surface of the SARS-CoV-2 virus, is primarily responsible for attachment of host cell and also the fusions of the viral and host cell membrane in order to infect us, right? This is a very, very important protein that allows the virus to infect us. So what we want, of course, is some sort of antibodies that can bind to the S protein so that it can block out the SARS-CoV-2 from interacting with these sort of receptors on the human cells. By doing that, we can actually render the virus unable to infect our cells. So this S protein, of course, S 抗原 is the most suitable target for antibodies to bind, right? Now that's great, but what makes antibodies though, right? I mean, we go out and do a vaccination, right? COVID-19 vaccination. Are we injecting antibodies? No, we don't, right? That's not how it works, right? So we need to know a little bit about, immunology background, right? So this is just cells of the immune system in a nutshell. Of course, this is very simplified, right, for kids. On the left side, we have the innate immune system that consists of at least 2 types of cells, which are the neutrophils and also the macrophages. And on the right side, we have the adaptive immune system, which consists of cytotoxic T cells, helper T cell, and also B cells, okay? So neutrophils and macrophage, they are like policemen in our body, right? They are phagocytic cells, means they can engulf, right, pathogens. And they are our first line of defense. They act very fast, but they have no memory, and their attacks are non-specific. Non-specific, meaning that they don't take any kind of special strategies to go against specific pathogens. They treat everything the same way, right? And they have no memory. Each time they attack, they use the same way to attack, okay? Now for the killer T cells, these are the cytotoxic T cells, sometimes we call them killer T cells. They don't actually directly attack the pathogens or viruses, right? They attack infected human cells, right? Our cells, after infection, becomes a factory, right, to produce more viruses. So killer T cells is responsible for killing these infected cells so that there will not be further productions of the virus, right? And then, of course, we have helper T cells. Of course, although these guys are called helper, they are not really just a helper, but they are the actual commander of the entire army, right? They communicate with B cells and the cytotoxic T cells and the macrophages to coordinate the whole attack on the enemies. And lastly, of course, we have B cells. They are actually our weapon specialists that produces antibodies. Aha, these are the ones that we are trying to talk about, right? Yeah, they are the ones that produce antibodies to neutralize the pathogens when they are under the command issued by the helper T cells. And most important of all, some of them possess long-term, long-living memory of previously fought pathogens, right? That will give us some sort of long-term protections. That's the beauty of the adaptive immune system. So if we look at right here, right, within 1 or 2 slides, I'm trying to conclude the whole immune system to you, right? So we have some sort of B cells here picking up, oh, I remember this guy, right, this pathogen, this virus, and I will make my antibodies so I can, we can actually combat against these sort of pathogens. As for the cytotoxic T cells, under the order of the helper T cells command, they will go and attack the infected cells so that there will not be further productions of virus in us, right? This is what we call humoral response, and this is what we call cell-mediated response. And after these things happened, right, finally, our innate immune system will come back and do the clean up the scene tricks, right? Basically scavenge, right? And clean up whatever dead bodies that are around so that we can go back to normal, right? So, of course, this is just for kids, right? But our immune system is pretty much like this, except it's much more complex, right? The ideas that are represented here, especially true, is, first of all, we have antibodies that are the humoral response, and we have cytotoxic T cells, the killer T cells, which are most of all our cell-mediated response. And both of these are the critical components of our adaptive immune system. Our innate system is important, but an innate system alone is incapable of subduing the virus, for example, like COVID-19, right? What we need is to train up our adaptive immune system so that they remember the enemy, so that they can be our most powerful weapon against the COVID-19 virus. Now, if we take a look at this charts right here, it represents the normal virus infections, the transitions from the innate to the adaptive immunity during this infection here. First of all, we start off with the innate immune response here that is responding to this rise of the viral load. So the amount of virus is increasing in our body. Then of course, your phagocytes, right, your neutrophils and macrophages will respond, and there are lots of responses right here. And then after a little while, they found that, oh, it's actually not enough to subdue the virus, right? And all these times, right, the adaptive system will start to kick in, right? Within 5 to 7 days, the cytotoxic T cells and antibodies will increase, right, in order to subdue the virus. So you can see how these T cells and antibodies increase and the viral load will go down. Like what we said before, adaptive system is our most powerful weapon. Now, if we take a look at COVID-19 infection right here, right, you can see how the innate response is not as strong as this one here at the beginning, which resulted in a much higher viral load being here. And then also the adaptive is kicking in much later. There is a delayed transition to the adaptive immune system, right? So by the time these start to kick in, it's already too late. The viral load is already too high, and the innate immune response, there's no chance for it to rest. It has to continue to buffer at this time before these are kicking in. So it has to go really strong. And so now the innate system then overreacts, right? It overloads, and it becomes a severe case of COVID-19 infection. So from here, we can see that, of course, what we want is we want to do some sort of vaccination, because vaccination trains up our adaptive immune system. our innate system, we can't do anything with it, right? Even after vaccination, they will still respond in exactly the same way as well. So no matter what you do, this is going to be like that anyway. But if we can actually have a faster, stronger adaptive immune response kicking in here, that will save us from having this severe COVID-19 case. And that's the beauty of vaccination. We can train our adaptive system using vaccination. So for COVID-19 vaccinations, of course, we have our well-known inactivated vaccine, which is usually this is inactivated vaccine is just having the virus itself gone through a procedure to inactivate with chemicals, heat, or radiations, rendering it unable to infect human cells. And the example of this would be Sinovac, CoronaVac, right? This is the classic example of inactivated COVID-19 vaccine. And then another choice that we have, of course, is the mRNA vaccine here. mRNA vaccine encodes the S protein, right, of the SARS-CoV-2 virus. And an example of this would be the BioNTech or Pfizer, right, Comirnaty COVID-19 vaccine. So how do the two vaccines work differently at the cellular level, though? First of all, let's talk about the inactivated vaccine. Here we have a person that just got injected with an inactivated COVID-19 vaccine, and this is that inactivated variant particle right here of the SARS-CoV-2. What happened is under our skin, we have a lot of these cells called dendritic cells, DC. They are not warriors. They don't fight. What they do is they engulf and they process these sort of particles and then presents the antigens to our T cells at lymph nodes. That's what a type of antigen presenting cells, APC, right? They engulf. So here we go, right? We engulf the vaccine, and then it's going to process it, take it apart, right? And present a part of it at the cell surface. And then it's going to run to the lymph nodes in our body. So it's going to deliver this antigen, this piece of antigen, as a military intel to the lymph nodes. And here we are having the lymph node right here. This is where our T cells and B cells are located, okay? And this antigen presenting cell, which is the dendritic cell, right, is going to go here and present this information and train up our army there, our specialists, T cells and B cells, our commanders and the weapon specialists there. And so now the armies in our barrack would get trained up, ready for battle, okay? So now this is a closer look at the lymph node here. Inside the barrack, right? This is the dendritic cell, and it's going to present this piece of antigens to a naive helper T cell. So this is our commander, but before he become a true commander, he's kind of naive, okay? Never seen an antigen. But after getting in touch with here, right? Getting the intel, the information, he will mature into the true commander of the army. Now, this is a mature helper T cell. Now, at the same time, at the lymph node, we also have B cells that we talked about before, right? These cells can also engulf and process this sort of vaccines, right? Treating it as a pathogen. And then it's going to present some of these at its cell surface using this MHC2 thing, right? We don't have to go into too details of these. And then what happens is the commander, right? The helper T cell would interact with this guy and turn him into a plasma cell. This is our real weapon specialist that can actually produce antibodies for us. This is the goal, right? To have antibodies. These are high affinity antibodies against the COVID-19, right? So, and while we are having these antibodies from these cells, some of the B cells that does not become plasma cell would actually become memory B cells. Okay, these are the memory B cells right here. They would actually express receptors that are very sensitive to the antigen. And so next time when they see the same thing, they would very quickly become plasma cells and produce a lot of antibodies. So here, for the antibody producing plasma cells, right, we created immediate protections from this inactivated vaccine, whereas these sort of memory B cells would give us long-term protection from this inactivated vaccine. Okay, so the advantages of the inactivated vaccine is that, first of all, it's very safe. It's a classic vaccination technology that has been around for many, many years. It is effective in inducing antibodies, right? The humoral response. It is easy for storage, but then the disadvantage is that, first of all, although there are good inductions of humoral response, there are no inductions of cell-mediated response at all. Although the helper T cells are activated by the APCs, which then trigger the B cells to produce antibodies, there are no actual infected cells to trigger the maturations of the killer T cells or cytotoxic T cells. Okay. So, and also another disadvantage is that inactivated vaccine, I mean, you pretty much take all those virus, right, and then inactivate them, right? You have no control over what part of the virus is going to give rise to the antibodies. There's not much specificity. So your antibody could be produced against the S protein. It could be produced against another protein in the virus, right? Could be inside, could be outside, could be anywhere. So although this can be a disadvantage, you may argue, hey, this can actually be an advantage as well. Because now you can actually, from this inactivated vaccine, generate many different types of multiple kinds of antibodies against different proteins of the SARS-CoV-2 virus, right? But it's a question mark. We don't know, right? It's an inactivated vaccine that's got into our body. We have no control over what protein is going to produce the antibody. Maybe you get some good ones, maybe you don't, right? What if you got a protein that is, you say, hey, but the S protein mutates a lot. When it mutates, right, if I have antibodies against another one, that would be nice, right? But what if the other protein is inside? So that's not really accessible by the antibody in the first place. Or maybe another protein that's just not responsible for infection at all. Then even if the antibody binds, it's not going to effectively protect the person. So yeah, kind of, most of the times, it's going to be the disadvantage more than an advantage in this case here. So, of course, we have another choice here, which is the mRNA vaccine. These vaccines work differently than the inactivated vaccine. First of all, it is the mRNA transcript that encodes a specific surface protein of the virus. In this case, of course, it's the surface S protein of the SARS-CoV-2 virus. And then what they do is they place the mRNA inside a lipid nanoparticle capsule so that it can be delivered into human cells, right? And it relies on our own cellular mechanisms to translate this sort of mRNA into S proteins inside our cells in order to produce the response. Now, this is how an mRNA vaccine works to protect us, right? So first of all, we have these sort of nanoparticles, lipid nanoparticles that holds the mRNA vaccine, mRNA inside. And after entering the cells, right, this sort of nanoparticle will help it enter into the endosomes. And then this will be released as an mRNA into the cytosol, where these sort of ribosomes will translate the mRNA into the viral S protein. Some of this S protein would actually be able to be secreted outside the cell. And then what happens? Once it's outside the cells, just like the inactivated vaccine, your dendritic cells sometimes can pick up these, right? And then it will result in the same fate as inactivated vaccine, right? It will take these to the lymph nodes and produce antibodies, right? Now, on the other hand, most of these would actually retain inside the cell. And what happens is they would actually go down a proteasome and becomes fragments, right? Become very pieces of peptides. And they will then go to the endoplasmic reticulum, where they would then be presented by some sort of MHC protein. So this is a kind of surface protein that is important, would take up a piece of these and present it outside the cell. This is what will happen in any nucleated cell in us, right? So what is this? What is going on here? Well, this is basically an ID check, right? In any kind of nucleated cell, we have a mechanism, right, in our cells to check whether this cell is infected or not. Usually, the MHC1 will present self-host protein, right, our own protein. And that is basically telling outside that, hey, you can check me, I'm okay, I'm valid, okay? But in the case where there is an infection, or in this case, there's a viral protein that gets processed here, then it's going to present this as a foreign protein. Then what happens is this is a death signal, right? Our cytotoxic T cells will be able to pick this up and find that, hey, you are not good. That's not a valid ID. So I'm going to kill you. So now the killer T cells will come in and carry out cell-mediated response, right, to actually kill things off. Now, on the other hand, apart from this side, that we have MHC1, which is presenting the antigen this way. If this is actually turned out, this cell is a dendritic cell or a macrophage, then what happens is that this would actually express MHC2, which is a different protein, which they would use to present the fragments again. And you know what? This is now the same scenario as this guy up here, right? If this is a dendritic cell and it has these sort of proteins in there and presents up here, it's the same as this guy presenting the things to the lymph node. So now this cell can present this fragment to helper T cells at the lymph node and carrying out activations of helper T cells, antibody productions, ultimately resulting from the B cell. So now we have both cell-mediated response and humoral response coming from mRNA vaccine. So the advantage of this clearly, of course, it fakes an infection, right? There's no actual infection taking place, but it fakes the infections. So now you have both humoral and cell-mediated response, right? Due to this fake infection. It is non-integrating. So unlike some of the rumors out there, mRNA cannot just incorporate into our genome. So our DNA is safe, right? Unless you have some special reverse transcriptase, it doesn't turn into DNA to be incorporated into our genome. So it's very safe. On the other hand, mRNA actually has a short half-life, which means that it doesn't last very long, right? It will naturally degrade very quickly, relatively quick. And then also high specificity and efficacy. And last of all, right, it's easy to design and modify as well, right? On the other hand, the disadvantage is that, first of all, it is not that stable. I mean, it's both an advantage and a disadvantage, right? So on the disadvantage is unstable, so you can't store them for too long. On the advantage is that because it's unstable, it doesn't last very long in us, right? So that we can get rid of it after a while. Now, at the same time, this is kind of expensive for storage then because of that, and transport. And also there are some sort of concerns about side effects and long-term safety. After all, this is still a new technology, so no one can guarantee what happens after, 10 years of use. Okay. So, but anyway, so this is very clear that we have both humoral and cell-mediated immunity, right? On the left side here, we talked about the presentations of the antigens by MHC2, right? Activation of helper T cells, B cells turning plasma cells, antibody production, which is humoral. On the right side here, we have like most of our cells that would actually activate cytotoxic T cells. Actually, the helper T cell will also take part in this, right? We don't want to get into too much details of that. But meanwhile, this will also happen on the destruction of infected cell, which is the cell-mediated immunity. Most importantly, now, actually, apart from memory B cells, there are actually also memory T cells in us. So apart from these guys giving rise to the memory B cells, the activations of cytotoxic T cells, sometimes they get inactivated after the infection is gone, and they become memory T cells, which would be much more faster to react to the next infections, right, to the same kinds of pathogens. So while these two offers immediate protection to us, these two offered long-term protection for us, right? Both memory B cells and memory T cells right here. So this is a picture showing you a comparison of the first exposure versus the second exposure of the same pathogen, right? Same virus. And this is the first time exposing to the virus, and this is your primary immune response of the adaptive immune system. This is not innate, right? This is the adaptive immune response. This curve here represents the concentrations of antibodies and the number of T cells in us, okay?